All skeletal muscle composed of numerous muscle fiber ranging from 10 to 80 micrometer in diameter. All fiber extends the entire length of muscle. Each muscle fiber contains several hundreds to several thousands myofibrils, and every myofibrils contain the light bands that contain only actin filaments and are called I band. The dark bands contain myosin filaments as well as the end of the actin filaments where they overlap the myosin and are called A bands. The portion of myofibrils that lie between two successive Z discs is called sarcomere. Side-by-side relationship between the myosin and actin filament is maintained by a large number of filamentous molecule of a protein called titin that is very springy, which holds the myosin and actin filaments in place. One end of the titin molecule is elastic and is attached to the Z disc. The other part of the titin molecule connect it to the myosin thick filaments. Spaces between the myofibrils are filled with intracellular fluid called sarcoplasm containing large quantity of the potassium, magnesium and phosphate and a tremendous amount of the mitochondria that supply energy in the form of ATP. Myosin molecule composed of 6 polypeptide chains that two heavy chains wrap spirally around each other to form a double helix which is called tail of the myosin molecule. One end of each of these chain is folded bilaterally into a globular polypeptide structure called myosin head. Thus, there are two free heads at the one end of the double helix myosin molecule. The 4 light chains are also part of the myosin head. Myosin filaments made up of 200 or more individual myosin molecules. The protruding arms and the heads together are called cross bridges. Each cross bridge at 2 points called hinges. Actin filaments are composed of actin, tropomyosin and troponin. The backbone of the actin protein molecule is double-stranded F-actin molecule that is composed of polymerized G-actin molecules. Attached to each G-actin molecule is one molecule of the ADP. These ADP molecules are believed to the active sites on the actin filaments with which the cross bridges of the myosin filaments interact to cause muscles contraction. Tropomyosin molecules, the active filaments also contain another protein called tropomyosin. These molecules wrapped spirally around the side of the F-actin helix. In resting state, the tropomyosin molecule lie on the top of the active site so that attraction cannot occur. There is additional protein molecule attached to the tropomyosin which is called troponin. It is complex of 3 loosely bound protein subunits. One of the subunits has a strong affinity for actin, another for tropomyosin and the third for calciamines. If the troponin-tropomyosin complex is added to the actin filaments, the binding between the myosin and the actin does not take place. So the active sites on the normal actin filaments of the relaxed muscles are inhibited by troponin-tropomyosin complex. In the presence of large amount of calciamines bind with troponin C molecule. The troponin complex positively undergoes a conformational changes that affect tropomyosin and moves it deeper into the groove between the two actin strands, which uncovers the active site of the actin, thus allowing these active sites to attract the myosin cross bridges head and cause contraction of the muscle.